Okay, so we've learned a lot about gene editing in the wild and how it might work, but we should dig into some details. So to start with, Jared, can you tell us a little bit about the human genome?
Yeah, absolutely. So when we talk about the human genome, we're talking about all of the genes that are in each human that make us who we are. And those might be a little different in each person, but everybody should have roughly the same genes in all of us. So how those genes are stored are in something called chromosomes, which you see here. So in each of your cells, you have 23 pairs of chromosomes for a total of 46 chromosomes. And the DNA is super, super tightly wound around proteins called histones, which we'll tell you about in a minute, to form these chromosome structures. So when we talk about the human genome, we're talking about all of the genes that make us human, and those are found in these chromosomes.
That's really cool, that DNA can pack itself super tight like that. So if it's packed really, really tightly, how do you actually get to a specific part to edit it?
That's a good question. I think first, if we talk a little bit more about that structure, it might be easier to visualize. So as you can see on the right here, we have one of the chromosomes that we were just talking about. And then if we zoom in even further, you can see how tightly wound it is. And over here we have the histones, which are those proteins that the DNA winds around very tightly. You can kind of think of that like a thread around a spool. So it organizes it, it makes it easy for the cell to move that DNA around, and it also makes it easy for the cell to access that DNA. So as you probably or maybe don't know, the DNA has this double helix structure, and it's able to maintain that structure while wrapped around the histones. And so when we're editing the gene, we're unraveling it just a little bit from the structure that you see here so that we can change the DNA.
That is super helpful, and I'm definitely glad we have an organizational structure. Otherwise, we'd just have DNA everywhere.
I know, that wouldn't be good, right?
Not at all. So, are there different options for gene editing, or is this like a one-size-kind-of-fits-all thing?
There are tons of options, and I'm glad you brought that up. We'll talk about one specific one here, though, that is one that's very common. You might have heard of it, but if you haven't, we'll tell you about it here. It's called Crispr. And CRISPR is super cool. It's a super common method that's used for gene editing right now. It was actually found by studying bacteria. So CRISPR refers to molecules that bacteria cells use as part of their immune system. And what they do is when a bacteria cell gets infected by a virus, it uses those molecules to cut up the virus's DNA into tiny pieces. And then, if the bacteria gets infected again with the same virus, it can recognize it a lot more easily and start its immune response.
Do they really cut it up with tiny scissors?
I wish they did, but I'm not quite sure about that.
I wish they did too. That would be adorable to think about tiny bacteria with their tiny scissors.
I agree. They'd be kind of just like us in that way. But what's cool about this is that researchers were actually able to take those tiny molecular scissors and those other molecules that are part of CRISPR and pull it out and use it in the lab to actually edit our DNA. So kind of like you see here, they can take their tiny molecular scissors, make cuts in that DNA which has been unwound from the histones, and then we're able to put in genes to repair different dysfunctions that our genes may have if we're sick.
Wow, I'm really glad that we have this technology, that it's been developed, and that we have a real scientist in-house to tell us about it.
Yeah, I mean, me too. So if we think back to like Connor, they might have used something like this to change his dystrophin gene to something that works properly for him.
That makes a lot of sense.
Yeah.
Well, thanks for following along today, and I hope you learned a little bit something more about personalized medicine and also gene editing, and that you got a little more excited about the possibilities of personalized medicine.
Yeah, me too. And if you're interested in learning more, there's a lot of resources you can check out online. You can go to the library. I'm sure you could ask your teacher more questions. And thanks for joining us.
Bye!
Bye!